The overall goal of this imaging protocol is to describe the quantification of intracellular pH using a genetically encoded pH indicator, and demonstrate how this method can be used to assess basolateral proton transport in an insect renal structure model, the Malpighian tubule.
This method can help answer key questions in the solute transport field, such as how specific transporter proteins contribute to epithelial proton movement and intracellular pH regulation. The main advantage of this technique is that solute transport can be quickly and easily assessed across multiple functionally distinct zones of intact epithelia. Though this method can provide insight into pH regulation in insect epithelia, it can also be applied to other systems, such as the mammalian nephron and epithelial cells in culture.
To begin this procedure, position 2 sets of soldering helping hands clamps on the imaging microscope stage with 1 clamp on each side. Next, insert the respective bent glass capillaries in the inflow line and the vacuum-connected outflow line. Then, mount them in the helping hands and align them with the imaging stage of the microscope. Spread vacuum grease onto the sealing tape and press the adhesive perfusion well divider onto the tape to coat the bottom with grease. Peel off the adhesive perfusion well divider and place it grease side down on top of a poly-L lysine coated slide. Following that, remove the perfusion well divider to leave individual specimen wells traced in hydrophobic grease. After that, place 200 microliters of room temperature